Hello, everyone. Welcome
Hi.
to the. Sorry. Excellent.
Hello.
Hello, everyone. Welcome
Hi.
to the. Sorry. Welcome to the Microbiology Journal Club. I'm Faz Alam. This is my co-host Danny Chan, and we're here to talk about microbiology.
Yes, we started up this journal club right as we were getting locked down. We used to do it several years ago on Twitter, and we still accept questions and so forth via that medium. So if you have any questions for us or suggestions for papers we should cover, please tweet us, add us either at our account or with the hashtag. Yeah,
Yeah, the hashtag, which is now situated below us because we've got a brand
ah,
new
it's
overlay.
changed.
Yeah, it's changed. Sorry, I changed everything now, so we look a bit more fancy. We've got a bit more neon going on.
Nice.
So trying to look a bit more like as good as those YouTube gamers, except we're talking about science.
Yeah.
Uh,
Yeah,
yeah,
that's our competition.
yeah. We're so this week we're covering a COVID paper on T cell immunity.
Hmm.
So yeah. So
Yeah, it was we should probably
so
say the title.
yeah. Titles for science paper.
SARS-CoV-2 specific T cell immunity in cases of COVID-19 and SARS and uninfected controls.
Yeah. So yeah,
Very
all science.
descriptive.
Yeah.
That's the right paper. That's what they did.
Yeah, that is pretty much exactly what they did. I mean, the link is in the description if you want to read along with us. So it's always available if you want to pause this video and download the paper and then scroll along as we're talking about it, so you can figure out what we're discussing.
Yeah. And so this is our on-week broadcast, and what we'll do is we'll go figure by figure. We have some questions that I guess, typically follow, like journal clubs would follow. We put
Hmm.
them online in our microbiology journal club, and it starts talking about, I guess, what people know. What have people done this? What have people done in the past around this topic?
Yeah.
Um, and I think the introduction is so short on this paper. It was like two paragraphs or something like that, and it was like talking about, set it up. Oh, SARS, it's a big deal. Lots of people are going down. Um, I think the most interesting was there were two papers they cited. One that was done in SARS and one that was in MERS,
Mm.
and it had shown. They did a similar thing where they looked at convalescent serum from people who had different forms of the disease, and they looked at the T cell profiles, and we'll
Mm.
get into that as we talk about this paper as well. And what they found, though, in those papers was there was some correlation between having a robust like T cell response to these antigens and less severe disease. Right. So there's some indication that this could have some clinical, just looking at SARS and MERS from the past. But I mean, they were a lot smaller sample sizes. At least the MERS one was much
Mm.
smaller, right? Because yeah, less
Yeah.
people got it. I think it was harder to get a cohort together. Um,
Yeah.
so like as with all science, like you gotta take those past everything gets taken with a grain of salt, right?
Mm.
Because like just because you saw that, just because this is the reason why they set out to do this paper, right, and like study T cell responses, doesn't mean that like it is gonna end up being the useful metric for looking at protective immunity.
Yeah, I mean it's interesting because like we've been looking through lots of papers, and they tend to on COVID-19 and vaccines and stuff, and they tend to focus on the antibody responses. But obviously,
Mhm.
there's this whole other aspect of the immune response related to T cells that are theoretically more geared towards viruses that tend
Right.
to like do all of their like metabolism inside of cells. And T
Yeah.
cells are designed to destroy like cells specifically infected with viruses. So
Right. Yeah.
having
And
that,
and I think the point there is it's this CD8 T
mm,
cells. And like
yes,
there's going to be tons of this, I think, during this discussion about this paper, because immunology is all about like these specific molecular subtypes of cells that go and do
yes,
things, right? Like we all have the story of or like maybe a
mm,
lot a lot of people might understand like immunity as this story of like a battle, right? Where
mm,
you have cells and they're surveilling for things, and then when they see something bad, they like tell their friends, and then their friends come and like make some molecules to like break up the party. And an important part of and one of the ways to think about all of those different cells that are going off and telling them is this CD8 CD4
yeah,
locus, right? So like you could be like if you have their receptors, right? CD4 I think is the T cell receptor basically,
Yeah,
right? And
so
they're both antigen specific, I think is important to say.
yeah, so
Uh,
CD8 and CD8 are what are called natural killers, and CD4 tend to be more T helper cells,
yeah.
and but
Natural killers,
obviously,
but all sorts of things. Like natural
yeah,
killers I think are a subset like
mm.
these are like very broad descriptions, right? Like
Oh yeah,
underneath the banner of CD4, there's like T helpers, like there's different types of T helpers. There's and under the banner of CD8, there's NK, there's cytotoxic,
yeah,
right? There's like a lot of different things,
yeah,
but the I guess one of the ways to think about it is CD4 is more. I guess I want to talk about the receptors themselves, right? Like
yeah,
CD4, they're both antigen specific, right? So
hmm
they both respond when they see a very specific antigen, a shape,
yeah,
right? And in CD4, they're going to engage that shape on other immune cells,
yes,
right?
that's
So
right.
I think that's an important thing to say. So like CD4, when you have a matching CD4 for the antigen, right?
Hmm.
What it's communicating with is another immune cell,
Yeah,
right? And so that could be a cell that was doing the surveillance, right? Being like, I found this, it looks suspicious, and then a CD4 would come in and touch that and say, okay, you think this is suspicious? I am the cell that holds the right that holds this specific antigen, and okay, I'm gonna now go and tell other people, right, that you said that this was a suspicious antigen.
yeah,
But on the CD8 side, those are engaging MHC molecules, which are
hmm.
on every cell.
Yeah,
Yeah, and so that's a bit different. So it's not. So now it's like you're a regular cell, and like something inside of you is going weird, right? And
yeah,
you show this antigen, right? And the CD8's going to touch that and say, oh, like you have the markers of something that is not going right, right? And so
yeah,
like there's the information flow is different, right, in these two
mm.
circumstances, right? That when we're thinking
Yeah,
about CD8 T cells, like that's very much like the body itself, the sort of other cells in the body, right? Something's wrong inside of me, or like they don't know what's wrong or right, right? Like something is going on inside of me, right?
hmm.
Does this do other cells in my environment have something to say about it,
Yeah,
right? Versus CD4, we're saying like you'll see these cells come up if, like that's like one of the branches to get into the antibody response, right? Is
yeah,
like um, who has seen this, right? If you've seen this, right, then we have to start a different cascade. We have to go down a different side.
yeah,
Um, and we'll be focused. I think, and so I guess what we want to talk about, or what we want to focus the discussion on this time, is T cell immunity. There's both these elements. One of them is actually like an effector side, right?
mm.
The CD8 side is very much an effector side,
Yeah,
right? They are doing the things when they see the antigen, they do the things.
yeah,
Versus CD4, it's going down the path of they are going to instigate a response, right? Also important
yes.
for the immune system. Yeah.
Yeah, they're a support class. They're very important. I mean, CD4 are the cells that are attacked by HIV, and which is why lots of the responses went. So, I mean,
Go
tends.
down. Yeah.
Yeah, and that's actually been like an interesting thing for learning in immunology because you learn a lot about a system by what happens when you take out one little part of it and seeing all the rest of it collapse.
Hmm.
And and so
Yeah,
it's
and I think that's that's I think we have a very uh, we look kindly on basic research in
mm,
this journal club, right? And
yes,
that's an important point to bring up that even if there's no and if there ends up being no clinical impact for the information that comes out of these studies, we are also learning about the immune system,
yeah,
right? In very strange, right? Because like viruses are these tiny little things that like muck everything up, and when we focus in with that laser focus and ask very specific questions, we end up finding interesting information about other stuff.
yeah, exactly.
Mhm.
So I mean, we've gone through some of the background of it.
Yeah, yeah. Let's
Talk
do the
about
figures.
let's do the figures. So what they did was I mean, figure one like talks about the actual proteins that they're looking at.
Mhm.
Um, so if I'm looking at figure 1A, so it focuses on the SARS-CoV like proteome. The all and from that they check out some specific proteins. So they decide to look into. So I'm going to be like jumping into one of the extended figures. So this So during this jump up, we'll be jumping in between extended figures. I try to line them up as they appear in the text.
Mm.
So just so we can like have that smooth reading order as we
Yeah,
go through it.
as if we
Um,
were following the reading of the paper.
yeah, and so it's very part of reading papers can be quite complicated because everything is out of order due to weird publishing restrictions, weird histories and stuff. But we're going to try and present the information as cogently as we can.
Mhm.
So they've basically highlighted most of the SARS-CoV-2 like protein and some of the main kind of like targets of it, and then they decided to look into like some specific proteins. So the nucleocapsid protein and the NSP7 and NSP13. So they go into a bit
Yeah.
more about this in extended
Yeah, so that's
figure.
the orange bar and the red bar and then the dark blue bar. They put little
Yeah.
asterisks besides the one that they're looking at. But they show us
Yes.
all the proteins there. And I just want to point out, like, that purple one, Spike, we've been talking about that one a lot in the
Yes.
past episodes. That's the one that a lot of vaccines are being developed around, right?
Mm.
So we could make antibodies against those things. But
Yeah.
they choose NP and NSP7 and NSP13 because they're made a lot. There's a lot of
Yeah.
it in the cells. Like NSP7 and 13, those are part of the replication complex, right?
Yeah.
The RNA dependent RNA polymerase, so to duplicate the genome. So those are some of the first proteins that are made inside of
Yeah.
an infection. And then the NP, that's the nucleocapsid or the nuclear protein, right? So
Yeah.
the RNA, we know it's an enveloped virus. I'm sure we from washing
Yep.
your hands, break that envelope. But
Yep.
there's also more coding. There's like protein coding around the genome as well.
Yeah.
And so that's the MP protein. And you can imagine there's a lot of that because,
Mm.
like in a sphere, right? Like,
Yeah, you need
have
to
the
like
genome.
have every bit go in. So you definitely need more of that than any other protein. So,
Yeah.
I mean, because coronavirus, it's weird because it doesn't just like most RNA viruses, it transcribes an RNA, but then a lot of them would just like transcribe that RNA over and over again. Whereas coronavirus transcribes these subgenomic RNAs that we've like kind of talked about before that are
Mhm.
signs of like replications, and so it's got this really complicated like process of creating itself that allows it to do things like managing more like capsid proteins and
Yeah.
allowing less wastage, I think. Um,
Yeah, it's interesting. I'm not sure. Like, I wonder whether there. Yeah, we know there's all these subgenomics. Like all the other proteins there, they get made not from like the original genome, right? Like
hmm,
they have to make like smaller pieces that
yeah,
make each protein. Yeah, maybe nucleocapsid could play a role in making sure the full genome gets packaged in. That's possible.
yeah. I mean, yeah. Sorry, I was kind of speaking a lot in generality,
Mhm.
generalities.
Yeah.
I was, but the idea I was trying to get across was that it regulates its production of these proteins in a way that you don't often see in viruses that are single-stranded. So,
Mm.
um, well, I say I haven't often seen, but that's because I
Oh
haven't really looked into
yeah. I'm
viruses.
not sure. I,
So,
I think they all have to. All viruses have to at some
yeah,
level
that's
that
right.
regulation. They don't all do it through subgenomic though, right?
Hmm.
Like it's not always that they make a smaller piece of the full genome to make the protein. Sometimes they have ways of like restricting where the ribosomes latch on the whole thing.
Yeah.
Sometimes they do it through proteases. So like for example, like
Hmm.
in ORF1, right, the one that makes the replicase, that comes out as a polyprotein, I think,
Hmm.
right? So that one comes out as a polyprotein, and then they need a protease to chop it up into pieces, right? And then that
Right.
comes together and makes the genome. So like some viruses do it like everything's a polyprotein, right? It all gets chopped
Yeah.
up and then the pieces. So like, anyways, there has to be control at some level, and coronavirus ends up doing it through these subgenomic methods. Yeah, but that's outside of the scope of what we're talking about today.
Yeah.
Just that, like, I just wanted to draw attention since we're looking at the figure of all the ORFs, right? Like what we've talked about before, like how it all plays a role in the infection cycle.
Yeah. And a thing that they also wanted to focus on was the homology between different species of SARS-CoV. So this brings us to the extended data, which extended figure 1 and 2, where they line up all the like kind of sequences for all the proteins. For like the related proteins in different versions of coronavirus. So
Yeah.
they compare SARS, SARS-CoV-2, MERS, and unfortunately it didn't come out to. The slide has very small writing,
Oh,
unfortunately. So
OC43, HKU1, NL63, and 229E. Those are all seasonal coronaviruses.
yes,
Yeah.
then they're less celebrity, less like kind of famous than the other ones, but they're still important because they circulate in the population, and it shows that there is some like relatedness in so the nucleocapsid protein, which is the biggest one. Like between like the big celebrities, that seems to be quite homologous, but then
Mhm.
less so with the seasonal ones. And
Mhm.
then NSP7 and NSP13, they're they're very similar across all of them, which is interesting, and I think that's why these.
I
Sorry.
was gonna say kind of I would say there's some sense to that, right? Because
Mm,
remember the nuclear capsid protein, I mean, as far as in the most general sense, its job is to coat the genome, right?
mm,
And make sure that it can get into the envelope, right?
yeah.
After when the things bud. But NSP 7 and 13, those are part of the RNA creation machinery, right?
Yeah.
And like making RNA is like that's a very complicated enzymatic process. You can imagine
Yeah.
that all some of the conservation between NSP 7 and 13 across all of these, like those are catalytic domains, right? Those are like driving binding, right? There's like
Yeah.
important things you have to conserve between these guys that maybe aren't so important in the nucleocapsid because the
Yeah.
nucleocapsid just has to like find the genome and put it in, right? Versus like,
Yeah.
oh, you got to like bring in the nucleotides and like weld them together and all
Yeah.
that sort of stuff in the
Y- y-
enzymatic
there
side.
isn't much patience for messing up in that a bit. You can't go like, oh, we got mutation. Oh, everything's in it backwards now. It's like, okay, fine. You need to kind of be very
Right.
much just on the ball with
Exactly.
that.
Exactly. There's like design constraints in the
Yeah.
evolutionary space, right, that are constraining NSP 7 and 13.
Yeah. Um.
Yeah, but I guess the takeaway though is that in our celebrity strains, right, between the SARS strains, there's a lot more conservation than between the seasonals, but there
Yeah.
still is conservation even in the seasonals in these proteins,
Yeah.
which makes sense. They're part of the same family.
Yeah. And so next like part is like figure 1b where they describe what they do to so they take some of these proteins and they chop them up into small 15 polymer length fragments, which is
Mhm.
kind of like the ideal length for a T cell receptor to hit because
Yeah.
they are
It's things that get loaded into the MHCs of the T cells,
yeah,
right? Like, yeah, it's like that's like the length things get chopped up in the cells, sort of, to be exposed. So that's what those receptors look at.
yeah, and they like
I guess wait, we should probably say like very briefly, like, so that's right. Like, this is about CD8 and CD4, right? Are both are both receptors for antigens,
mm,
right? So like, I guess you could imagine them as antibodies stuck on the outside of cells in some
yeah,
ways. They're not the shape of antibodies at all. They
yeah,
have something totally different. But
yeah,
like they're the sticky end, right? They're the
yeah,
things that are sticking to things. But
yeah,
then on the other side is antigen presentation,
mm,
right? And so that's the MHC side of things. And in that sense, it's like inside of cells, proteins that are inside, if they get degraded, they get chopped
mm.
up and they get loaded onto presentation platforms, right? And those get put on the outside of the cells, and that's MHC. So when they're using these peptides, 15-mer peptides, that's about the size that would be presented on an MHC molecule, right? So
Yeah,
that's those receptors like to look at that size.
yeah, and they chop them up into small, and they have them in various pools. And in those pools, they do this thing called an ELISpot, which is where they uh, present these like peptides and add some T cells and see what like pops up. I think
Yep.
what grows on them. Yeah, and
Yeah, we talked about this one before. It's like each dot on the spot is presumably a cell, right? That
yeah,
has responded to that peptide and now is making some molecules that say I'm on.
yeah,
And they're using interferon as the molecule that says that they're turned on. And
yeah,
the reason is because interferon is like an important cytokine that's used
yeah,
in like all this antiviral signaling, and it's well known that this is the on signal for these types of cells, both CD4 and CD8
yeah.
positive.
And I guess we can move on to like figure C and D are both kind of presented together in the paper.
Mm-hmm.
Uh, So this is this is interesting because they like go into the COVID-19 recovered patients and look at which kind of what do other T cells responding to, which kind of fragments are these proteins coming from, and it tends to be so it seems to be mostly the nuclear capsid, like the sorry NP1 and NP2, right?
Hmm.
And that
Yeah,
seems
those are nuclear capsid.
yeah, that's right. I need to
Or
double check because I've realized
nuclear protein, I guess, is why it's
yeah
NP, but capsid
yeah
is fine.
I just realized that there's like if there's a drink game, every time I make a mistake, someone is going to get very drunk, like because I've run through, I've made so many mistakes. So anyway, yeah, but and it's interesting because the like NSPs are they're there, but they're not immunodominant, as I guess I think the term is, where one kind
Hmm.
of antigen dominates the whole immune response.
Hmm.
Um, and they show the data for. I mean,
Yeah.
they
And these are in COVID-19 recovered patients, right? This
yes,
is the blood from COVID-19 recovered patients. They took out the cells, they exposed them to these peptides. This is what they end up seeing
yeah, and they
in
show
terms
it
of
for
response.
each individual patient. So there isn't really any statistics to be had here because they're showing each individual, so you yourself can see the like variation in the
Hmm.
population that they've sampled.
Hmm.
So
Yeah,
um,
almost all of the patients have some sort of response to
yeah,
the nuclear protein, but only a few of them have responses to the non-structural proteins, the
yeah.
replicates complex.
So now we're going to get into the flow cytometry section, or as we
Yeah.
know, there's the facts of life. So um, yeah, so I'm going to start off with an extended figure, so where they show like how they can how. So this is a good way to like kind of figure out how flow cytometry works. So uh, they show you like the kind of the raw blood sample coming through in like panel A, where they check for scattering like in so forward scattering for area and for height to figure out essentially like it doesn't exactly tell you the like kind of size of cells, but it gives you a good indication of how
Yeah.
big, what kind of size range you've got your cells in, and then from that kind of section, they get the. So I mean, for people who don't know what flow cytometry is,
Yeah. You definitely have to
yeah,
say that.
I should have I muddle everything around again as I do, but yeah, so
No, you're good.
yeah, okay, so you have like a laser that kind of that that well at least one laser that goes through cells and like you get this like kind of funnel that you pours through cells individually and a laser to like kind of go through them and then on the end there's a sensor that shows that basically like kind of measures how much the light is like scattered in like the forward direction, the side direction, and it can also measure like the colors of the cells. So if you've got like
Mhm.
a dye on the cells, it can measure like what color it's coming out of. So you can have
Right.
different multicolored lasers measuring things. So
Yeah.
you can measure
Yeah,
like
so yeah, so in the earlier so ELISpot, right, is measuring like are the cells making this, right? Every cell like lands on a plate, and in that spot on the plate it makes some molecules, and then we can develop the color on them to say that that one cell made a thing.
yeah,
And so this is like another way of getting at that, right?
yeah,
This time the cells are going through that funnel, single file cells through a funnel, and each one gets interrogated by a laser.
yeah. Because we can't tell what like cells are creating things. So the only way we can do that is to try and find out what mix of like proteins they're making on their surface or internally. So this is a way to detect whether they have the CD8 antigen or CD4 antigen. So you add an antibody that's glowing in that certain color or has a fluorescent tag that shines when you put a laser on it. You can tell, oh, this one's shining red because I put a red tag. It must be a CD4 because that, and from there you can kind of differentiate which cells. But even before that, you have to differentiate that what you're looking at is actual cell because sometimes those antibodies stick to random debris, and you need to make sure
Yeah.
you filter that out.
Yeah, I mean, in the way that they're using it in E, they're using it exactly the same as the ELISpot, right? This is a
Mm.
confirmatory, it's another technique that's giving us the same information essentially. Well,
Yeah,
they're going to give us a lot more information as well, but like similar because they're also staining for interferon gamma, right? Like at the end goal of this is like we want to see again, right? Can
yeah,
we see that there are cells that have interferon gamma production when they're exposed to these peptides?
yeah,
But yeah, as you said, like in this format, all the cells are getting mashed up and going through a single funnel, but that's like all the cells that you've taken out in the
yeah,
blood, right? Like that includes a lot of stuff. There's like crud, like as you said, size things that clump together, right?
platelets,
Dead
there's
stuff is going to be
also,
small.
yeah.
Yeah, platelets, all sorts of things. Like it's messy, um, and yeah, they have to do these what they call gating
Mm.
strategies in order to exclude the stuff that you don't want to see.
Mm.
Yeah, so yeah, A is forward scatter and side scatter, so that's for the size, right?
Yeah.
Because big stuff is gonna scatter in one of the directions more than the other,
Yeah, so
right?
in terms
They're
of
not like
refraction, yeah, it's going to like
Mhm.
bend light a lot more because the light is slowed down a lot more, so the refraction angle is going to be a bit off in more different directions.
Yeah,
And
like if you imagine like a rectangle tumbling, right?
yeah,
A rectangle tumbling around and you shine a light on it, right? The size of that shadow is getting big and small, right?
mm,
As the rectangle moves around, right? And so like that's like, oh, okay, so that's like a weird shape, you know? Like
yeah,
I don't want that. I want a cell. A cell should be like a little square, right? A
yeah,
square as it tumbles around and the light goes. That shadow is going to be a more consistent size, and that's like, oh, that's a single cell. I want that. So that's the forward scatter, side scatter thing.
yeah. And
Um,
well, side scatter is for the stuff inside
oh,
of it because
right.
when a cell's got lots of stuff inside of it, the light can go in all sorts of directions. Because cells, we kind of like to think of them as big, transparent blobs, but actually some of them have some like nuclei in them. And white
Mhm.
cells have quite a lot of stuff in them because they have to pump out lots of protein and very quickly, and so they've got like a lot of granularity. So
Right.
side scatter captures that granularity. So the more granular the cells are, the more likely that they're lymphocytes. And
Right.
then
Yeah, sorry. Yeah, I misspoke. Yeah, that's the forward scatter. The shadow analogy is good for the forward scatter.
yes,
Side scatter is to think about what's inside.
yeah,
The light passing through those cells. So now, like that region, well, it's not really a shadow because they're actually transparent to light,
yeah,
right? And yeah, this is a good explanation. So that's why they have lymphocytes on the B gating, right? Because
yep,
like these are things that have a lot of stuff inside of them. It's not just
yeah,
nuclei. It's also like right. It's like all the granules, the effector granules and
yeah,
stuff that's going on. They're full of all the interleukins, right? They're full of
yeah,
enzymes and molecules that like they're just ready to go because in the immune system, there's a lot of chemical communication.
yes.
Yeah,
And yeah,
and
this is
you
interesting.
can't spend all your time like making that stuff, right? A lot of it's like pre-made, ready to go.
Yeah, exactly. They're like bags of just proteins waiting to burst and like fill
Right.
us with antibodies.
With the right signals, right? That's like
Yeah,
the whole complexity of all of this.
yeah,
So then they use like a live dead stain, right?
yeah,
Per CP. Um, and so they use two, right? One that is if it gets in, it's dead,
yep,
right? And then the opposite, if it gets in, it's alive.
yeah. So negative staining, which is great because then you can confirm like both ways. So you're definitely sure that
Mhm.
because sometimes because anything in biology there's always like a region where of like meh, it may work, who knows. So you just want to
Yep.
make that double confirmation, and
Mhm.
and so once you've got that, you've filtered out all those populations. You've got these two kind of relatively distinct populations of CD8 and CD4 cells.
Oh yeah. Well, then they'd use another laser, right, to look at
Yeah,
CD8 and CD4, right? So then it's
yeah,
like then they have to now there's an antibody that attaches to either CD8 or CD4 and has a color, right? So
yeah,
like you have to imagine all of this is quite fascinating from the technological standpoint, right? We went from a mashed up version of mashed up, no, not mashed up, it's blood, right? We've
yeah,
gone from like this like centrifuge down sample of blood passed through a tiny funnel, right, where we looked at the light that passed through it, the light that was like around it, the whether or not it took up different stains for live or
yeah,
dead, right? And then whether or not it had antibodies that also like had a marker for these specific molecular things, as well as the interferon gamma and the TNF alpha. We have antibodies for those as well,
yep,
right? And attached to that,
yeah,
and
yeah. So
and
we've
you end up getting these graphs. So like this gating strategy is from the initial stream of cells all the way down to the exact cells that we're interested in.
mm,
Yeah.
yeah, going straight from like physics to the biology aspect of things, which
Hmm.
is uh, always fascinating. But now we can see because I mean, when you look at this effects plot, which we're going to look at in a bit, it's nice to appreciate that there is a lot of filtering going on and a lot of like, uh, so it's
Yeah,
quite
you're only seeing the last filter, right? The
yeah,
filter that's relevant to our biological discussion, right? Like which are these which are these cells. But the people at the bench, they have to do a lot of different optimizations on their protocol to make sure that they're getting, what is expected at each gating step, right? Like
Yeah.
that the cells are healthy and that like these are actually lymphocytes. Like all of that has been taken care of before, and we get to see that final result in front of us.
And it's the research here presented because one of the key things is if you show your gating pulse, it shows confidence in your results because it is possible to like show some spurious data just because you accidentally gated the wrong thing. So there are so many nightmare stories from like 10 years ago where people finding new cell types that turned out to be because of people incorrectly gating things or not quite understanding how the equipment works, and suddenly we've discovered these whole new types of memory cells. And
absolutely.
So it's part of it. It's like from a normal immunologist point of view, this is actually kind of very dull workaday stuff, but it's actually kind of important for like kind of seeing how things progress and understanding.
And for other immunologists to trust your work, they would ideally want to dig down into this, right? And like we can only say like what we know that these things are, but an experienced immunologist would go in and say like, like I can tell that from this sample it looks like this. And remember, this is just an example. Every flow graph that we see will have all these associated gating graphs along with them, right? That data was all collected as well.
Yeah. So I guess now.
So E, we can talk about E then, right? So it's the same samples as they went through ELISpot. Now they're putting through flow, right?
Yeah.
And to look for the same stuff. That in the unstimulated you don't get anything, right? So that's the same as these negative controls in B, right? Empty ELISpots. But then when they add in those peptide pools, they start to see some of those cells shift over, right? And you can see they split into quadrants, and then they put little numbers in each quadrant to tell you what percentage of all the cells that you can see on the graph are in those different quadrants, right? And the idea is that there's two axes of signal. One is the CD8 axis. So from the bottom, there's no CD8 signal. As you go to the top, you're getting more CD8 signal. And then the interferon, the axes, that's like from the left, you're no interferon signal. Going to the right, interferon signal. So we're interested in stuff. The positive signal in this sense is going to be our top right corner, right? Because those are things that are both positive for the marker of interest, CD8 or CD4, and also positive for the cytokine of interest, interferon gamma or TNF alpha, which is another pro-inflammatory signal of like they're ready to do something.
And again, you've got this multiple lines of information. So they both kind of are indicators of information, but you want to check for both of them just to make sure that you've got multiple lines of evidence, which that strengthens your argument when you're presenting something like this in science. And they do show. I mean, again, the thing with facts is like they're often very subtle effects because number of population. Because if we're looking at people who even have a disease, the number of these populations is going to be very, very small. And these are counting individual cells from an individual blood sample, which is again a small sample of a much bigger population. So so even here, seeing like these like kind of
remember, from blood, but only a specific type of cell from the blood.
Exactly.
Right? Like
So I mean,
so like things like in E, subject C14, right? The TNF alpha graph, right? Like that's a really small effect. You're seeing unstimulated, there's 0.03% of the stuff in there, right? And then you're moving to 0.4, but like it's an order of magnitude, right? And that is considered positive.
I mean, because again, each of those dots represents a real cell. This isn't like going to be indirect data. We know that those cells existed because we actually have them, and they gave us that data.
Subject C14.
I'm
just trying to match it up. NP2. Oh yeah, okay. Anyways, all that to say, I believe they said that they confirmed all of their data from C and D using flow graphs, and E is just like an example of what they've shown.
Right. And so they've got like flow.
2 out of 7. I see. They did 7. They did 7 of the patients from the panel in C and D, and they're showing 2 of the graphs from there.
So they've got like so in like extended data figure 4, I think they show like a lot more of the patients, and I think that so again this is more to do with like the recovered COVID. So again, it looks at like TNF alpha. So cells that have like interferon gamma or TNF alpha or both
Hmm.
in different samples from patients against the different like nuclear proteins.
Yeah,
So
so this
they,
is this is a summary of all the graphs actually, right? Like
yeah,
of the seven that they did by flow, they now arrayed it in this bar graph style,
yes,
right? Yeah.
yeah. So I mean that's pretty cool data to have. It's so it does like highlight their argument that there are that the cells that are reacting are from these two subsets.
Yeah.
Um,
But they're heterogeneous, right? It's
yes.
not that there's like, oh, this response is always seen in CD4, IFN, gamma positive, right? It's
Hmm.
like, no, there's all sorts. There's double positive CD8s, there's TNF alpha responsive only, right? These are
Yeah.
very rough. These are markers of activation, these two cytokines, IF interferon gamma and TNF alpha. Yeah, and it could be like, when did these people get infected? There's like a lot of factors that play into this, right?
Yeah.
Like which ones are coming up positive. It could even be handling at some level as well. Like I'm not really sure how
Mm.
sensitive these cells are in terms of going down these paths, but yeah.
Yeah. Because there is quite a lot of variation in the responses they log. But I mean, then again, that's not necessarily to be unexpected because the way the immune system works, that
Right.
everybody's immune response is it's very hard to.
Yeah.
Often
Just
things are
that
very
just that
different.
we don't know anything more specific than, you're right, like these cells are responding from the CD8 and CD4 pools.
Yeah. Uh, yeah. So I mean, they look at so they basically they basically demonstrate the T cell response in SARS recovered patients, but then they go into patients who've been previously infected with the SARS-CoV-1 original
Hmm.
flavor. Uh,
And that's
so
two, right? Figure
that's
two.
oh, wait a minute. I think I've completely
Oh
glossed
no.
over something very important. Sorry. They also like looked at the outcomes in the SARS-CoV patients as well. Um, so I'm going to be talking about extended data figure 5 because I think
Hmm.
there was yeah because again this is very glossed over in the paper unfortunately, but this is quite important because they're trying to correlate the actual T cell responses to patient outcomes
Hmm.
and antibody responses. So this is quite an important thing to look at in the paper, even though it's been relegated to the extended data because I imagine it didn't like come out exactly how everyone hoped it would.
Well, this was again. This is when I was referring to those two papers where they did it with MERS and SARS. They did end up finding some correlation, right, between the severity of disease and the T cell responses. So this is sort of the natural. Right? They want to do it as well, but they didn't find as strong a correlation.
Yeah.
And it could be because they have tons of mild cases here versus moderate and severe.
I mean,
Uh,
another thing that is they are focusing solely on nucleic capsid and NSP7 and 13, so they don't actually touch spike protein at all. Whereas I think the other
mhm.
studies did look at spike protein,
Yes,
and
yeah, they looked at all sorts of proteins actually. Hmm.
yeah, so that could be it. It could be because they're very focused on the proteins that have high homology between different populations, and the
Hmm.
spike protein. I think we looked at a paper where they were quite different. So
And I think actually it's because like the way when I think about this paper as a whole, the fact that they went for nucleocapsid and these and the NSP7, NSP13, and their rationale saying of like talking about conservation
mhm.
between the different types, because ultimately like they didn't want to study just COVID-19 recovered people,
Mm.
right? They wanted to understand more, right? Like the cross reactivity, right, against people with regular SARS and seasonal, right? To
Yeah,
have some sort of comment to talk about like, um, if we can see these responses in these other types, right? What does that mean for protective immunity, right? What does
yeah.
that mean for the immune response as a whole, right? Like, is this the immune response that's giving us protection, or is this just like the immune response that happens, right? Like in a variety of different circumstances. So,
So yeah, I mean, this is quite interesting, and that's the great thing about that kind of basic science because it isn't just looking at this one situation. It's building up our knowledge base so that we can better respond to the next time we encounter a
yeah, like I think
coronavirus.
the point of the paper is a bit more, yeah, is like kind of a bit more pedantic and like, um, basic than this idea of like, is this the protective response for us? But of course, because they did these tests in human samples, like, do the analysis, right? Like I wanted to see this information for sure, like in Figure 5.
And I think another important thing to consider with this figure is they use SVNT. So these aren't actually, so in terms of neutralizing assays, they don't actually use real like coronavirus. What they do is they use like a version of spike protein and to see whether the antibody responses in these patients prevented that. But but as we've kind of seen, the responses with these T cells isn't necessarily acting on the same thing the antibodies would.
Well, I mean, in 5A, it's weird, right? Like they're looking at correlation between the T cell response and the neutralizing antibody response.
Yeah.
Which are they're a little bit different because, well, I guess CD4 presumably should correlate, but not necessarily CD8 because they're not the antibody makers necessarily, right? I mean, like, I guess like you can have effects where I think like the CD8 cells kill things, and then that makes more antigens, like because the cells are dead now, then they get surveilled by the immune surveillance, and then that leads to more antibodies because like there's more examples of like surveillance moments of like this is a bad thing. But yeah, I mean, again, I think that the reason this graph exists is because it was what was done in these previous studies, right?
Yeah.
To be like this, and they showed correlation in some of these circumstances.
Yeah. I mean, I just feel like the measurement they kind of use for it is already handicapping them in trying to find the response, because it's looking at like spike binding to a receptor, and that's not and if they were using a live virus, maybe they'd actually have a very different kind of result, because the antibody response might not just be so these cells might so I'm
But
not sure.
I mean, I think this is like a interesting, like I think there's another protein on the outside of the surface of the envelope,
Yeah.
right? But like these neutralizing antibody tests, like that is like you want things on the outside. So like again, yeah, I think that's another reason why A is very strange to think about mechanistically from the immune perspective, right? That these cells are like they're looking for things that are being surveilled on the inside, and then they're correlating that against, right, antibodies for the outside of a cell.
Yeah.
But again, it's been seen before, just not seen here. Just not seen here.
I mean, so I mean, I think the thing is, like, one of the great things about this paper is they actually produce a review reports alongside it. So the story of this figure is actually quite interesting, because what this was requested by the reviewers quite reasonably, that they actually wanted to see whether, because this is the first thing you want to see. Like, if you're interested in T cell response, you want to think, well, how does this relate to me? How does it relate to all the people who are in hospital? Does this is this actually going to be a good signifier of how well people have been dealing with the infection?
Right.
And they do kind of like see some hints of things in so I guess in the mild, moderate, and severe graphs. I mean, again, it's very difficult to interpret because, of course, the numbers between those graphs are different. So I'm looking at C, where you've got lots of mild, few moderate, and a few severe, and so it looks like there's something happening there, but it's quite difficult because of this statistical thing where the smaller population, the more likely you're going to see some giant variation in there. So
yeah.
So
yeah, I feel like there's a bit of a wash here because I don't really know what the conclusions you can get from some of this stuff is, except that there isn't any strong signal.
Yeah, I feel like there isn't a strong signal, and again, with you're looking at convalescent patients, people have had the disease, and like, so I mean, and some of them had severe. So, I mean, can we argue that these people are good at fighting coronavirus? I'm not sure.
Yeah, no, I think. Oh, I see.
Yeah.
Oh, yeah. Yeah, like maybe these people, that's not the right response you want, right? Because they ended up in that bucket of bad disease.
So, I mean, that's like kind of beyond the scope of this paper to.
So that's
discuss.
actually, I think that was something that was reported in the MERS paper. Was that there's like, it's like you want CD8 was not a productive type of immunity. Right? There was like you want to see like, oh, in one, this is the response, and in the other disease set, this is the response, right? You want to see those two different, like, that's what they're looking for in these graphs, but they don't quite find that.
Yeah. Yeah, but I understand exactly why they did it, and I think that it's good that they did try to take a look at it.
Oh
And
no, I'm excited
yeah,
that it was. And again, like I think that the reason why these original authors did it, like hearing that it was also added by the reviewers,
hmm,
it's like the original authors weren't really thinking that much about its relevance to disease and
yeah,
like and like what we're seeing in people. Like I think they were very much just like it's been done before in these, right? Like we have the tools to do it, and it's an interesting question, right? Like what would happen in these circumstances, and they went for it. And so like the reviewers helped shape it even more to have like a more clinical like kind of like outcome, which I mean again like I was really excited to see extended figure 5 as well, and I wanted to learn more about like the reasoning behind it, but it's not really discussed in the text of the of the paper.
yeah. But I mean, this is one of those rare papers that has actually got really good and insightful reviewers.
Mm,
So it's definitely worth like taking a look. Even though a lot of the things that are kind of said by the authors, but in of course very compressed format because the paper has to have very tight word counts. Um, so it is easy for because I nearly missed this even though I've read it already. Um, but
mhm,
yeah, so going into figure two, which
mm.
which so let me double check what this was. But so
So
yeah,
now they're
this,
gonna go deeper right into the epitope mapping for
yeah,
MP. They want to get specific. Like they had these big pools. They had two pools, MP1 and MP2, but that's like made of tons of different peptides, right, spanning the length of NP. So now they want to see like specifically which part of NP does the stimulation.
yeah, and that's because this is great because again we saw those maps of like how which parts of these proteins are similar to each other.
Hmm.
So this is actually quite an interesting thing to look at to see like
Sure.
which were so
But you can see it's not clear. There's not a clear consensus that comes out from this.
no, not entirely. Because again, it's very varies depending on the people you're looking at.
Yeah, because it's
Uh,
people. Because what we're doing in our immune system is this selection experiment, right?
yeah,
Where it's like we're hoping that our bodies have this variant, right, that matches the antigen in question, and different people will have different sort of amounts of that.
yeah,
Yeah. But we looked at this style of epitope mapping in the receptor. We did one about spike protein, right, where
yes,
they
and the
were looking
MERS
at. Mhm.
another like paper that was correlating like spike proteins of MERS, SARS, and the
Yeah.
latest COVID-19. So
Yeah,
so
exactly.
yeah, we've
So similar idea that just like the pools got smaller now, right? Smaller pools of epitopes that are in specific regions, and they use the Ellis spot as the readout, and they double check
yeah,
um, with B. They double check in cells,
yep,
and I think there's an important thing that they're going to start flipping between now: in vitro expansion versus ex vivo.
yes,
Um, and as we said before, like we're looking at really tiny populations of cells and blood, and you only have so much blood to go around.
mm,
And so, because in this experiment they had to do this huge panel, right, of all these different things, they actually had to expand the cells that they had in the blood.
yeah,
And that's done. And so, immune cells they do expand, right, in our bodies. The idea is that when cells like encounter right like the right signals that tell them that you're the one that we need
yeah,
we need more of you, they'll replicate, and scientists know the signal that requires, and so they can do experiments where they take out the cells that they think are the ones that they're interested in, and they just add the signal. It's IL-2 in this
yeah,
case, and then you get an expansion. So but again there's that level of it's not exactly the cells that
yeah,
right, because in the body that's a microenvironment thing, that's like one
mm,
cell touching another cell being
yeah,
activated, right, and continues to that. But in the scientist sense, like we did flow basically, and we
yeah,
separated in a flow graph the cells and we treated them with IL-2. So there could be things that change in that expansion.
yeah,
Yeah, and also in expansion, like some things are more competitively expanding than others. There's so much that's not really quite known in that space. So just to say that things that are done in vitro, they have with in vitro expanded cells, they have another layer of like uncertainty added to them,
mm,
right? Because like we're not looking at ex vivo, we're not looking at the cells that exist in the body at
yeah.
that moment.
We're
Yeah.
looking at their crazy descendants that are raised in petri dishes and don't quite know what a human body looks like. So
Exactly. Yeah. Exactly.
yeah,
Exactly. Those cousins.
yeah. So So yeah, that's something to always take into account with any in vitro thing, because again, once you take cells out of the body, they do change slightly. Maybe not much, depending on what. It always depends on what you're looking at and what you want to take away from them.
Hmm. Hmm.
So
But in this case, like what they're hoping is that their expansion gave them more cells with the same CD8, CD4 receptors on the outside, right?
mm,
So as long as that's the case, even if they don't respond in the same way, right?
yeah,
That they should be useful for this mapping technique, right? And they should be able to help us get at the answer: what part of nucleopeptide are these cells responding to?
yeah,
Yeah.
because I mean,
So I'm
yeah.
I'm okay with the use of in vitro cells in this way.
Yeah, I think for this specific purpose, I think that's a very useful thing to do, because we wouldn't get a readout as big or as like efficient without the proliferation, and you couldn't get cells
Yeah.
proliferating if they didn't already have that specific like receptor. So
Right, right. And again, like they also like they're looking at interferon gamma. They're not like going downstream and saying like,
mm,
now tell me about more cell signaling. Like tell
mm,
me how these cells respond, right? Like if they were doing those, I would say, I don't trust it anymore because like
yeah,
you've gone through a passage where like things could have changed. But they're only looking for specificity against that antigen on NP. So like, okay, fine.
yeah,
This is a good use of it. Yeah.
yeah. And so I guess figure two. Going up to figure three, I believe now.
Hmm. Yeah.
Yeah,
Figure
where
three.
so now they're going into like the original version SARS,
Hmm.
SARS-CoV-1 as it's now called, uh,
Hmm.
where where they now like, because nobody wanted a sequel to SARS, but now we have to
Yeah.
call it that, like the PS1. I mean, anyway. Yeah, so they do the similar thing as they did with the previous paper. They make a overlapping peptide library, and
Hmm.
then so I guess I'm going to jump straight to B, where they look at the epitopes of the immunodominant like
Hmm.
proteins. So
Yep. And here even more, it's all MP1.
yeah, yeah, that is actually quite interesting, because as we've said earlier, NP1 it varies a lot.
Hmm.
I mean, it has a lot more room to vary compared to the NSP7,
It
and
does, yeah.
so
But I think the point that they do try to make in the paper is like there's also a lot more of it. So maybe
yeah,
that's the reason. Like there's just so much of it inside of a cell. Because remember, like in this instance of like activating CD4, CD8 cells, it's all based on that antigen presentation,
mm,
which is done by cells that take up particles. Like they just end up taking up more of this protein and
yeah.
presenting it.
I mean, I guess this is because having more of it means that you're more sensitive to. So because there's more of it
Hmm.
around, there's more chance of T cells actually expanding based on of that, compared to the other ones, which are much earlier in the infection, so they might
Yeah,
not be there when there's any damage to cause like T cells to think there's something wrong.
right.
So um, that does
Yeah,
make
but it's cross-reactive. Like that's
yeah,
I do think that the surprise I do agree with you. It's surprising that there's so much cross-reactivity between these, considering the proteins are so different.
yeah, uh,
There are also biases in
mm,
MHC loading, right? Like
yeah,
not everything gets loaded into MHC, so
yeah,
it could also be driven from that side of it. That like this nucleocapsid ends up getting no one know. I mean, not no one knows. It's still being determined the full extent of like what types of things make it to antigen presentation, right?
yeah.
Like how does a cell decide out of an entire big protein what pieces to take out and which ones to show? And again, it's like it's not just showing it because there's co-receptors that get shown
Yeah.
at the same time to have the activation. So it could be something in that process as well that drives this,
I
right?
mean,
That
yeah.
like the regions that are similar between SARS original and SARS classic and SARS-CoV-2, like those get antigen presented in the same way, right? I
Yeah.
could imagine something like that.
I mean, it's interesting because I think if from a virus perspective, on a whole evolutionary scale, it's actually beneficial for them to present the nucleocapsid over other things, because that's more variable,
Mm,
so they can evolve out of it. So I
sure.
think there is like some sense.
Or it could be like I also think that it could be a noise. Like maybe
Yeah.
you want to present it because like what does it matter that you could detect it at this point? Because
Hmm.
by the time nucleocapsid's being made, those cells have already made all the genome. They're ready to bud out.
Yeah.
Right? Like it could be that nucleocapsid is not the right response here, because it's too late.
Yeah.
Once you figured it out, like, oh, it's too late. Like you can kill that cell, but it's already made a bunch of virus.
Yeah.
right?
Ah,
Like because remember, like the NSPs, those are very early
yeah,
produced, right? Those are going to be
yeah.
very rare signals. Like at that point, it's still up in the air whether or not the virus is going to be successfully made. Like it's hard to say. There's no again, like this is where I feel like this paper is so basic research style, right?
Mm,
It's giving us tons of fodder to talk about why we might think one way
yeah.
or right, like to develop hypotheses and go to the next step. Yeah, I really like it because it's also humans, and like
Yeah.
I feel like we're getting at something that is a lot more. Yeah, it feels very relevant.
Yeah, and it's that's I mean, oh yeah, I guess we'll move into number C. Sorry, figure C and D. Sorry, C and D in this where this one like again they're looking at ex vivo expansion, I think,
hmm
specifically. So
no, C and
C and
if you're
D.
yeah,
Uh,
no, C and D, this is the same. These are the matching figures from the Ella spot,
mm,
right? Looking at what's immunodominant. I've been talking about
yes.
C and D this whole time in
Oh,
some
good
ways.
grief. Sorry. Yeah. No, you because B was showing something similar. Sorry. Yeah. No.
Yeah. Yeah, I've been
Yeah.
talking
No.
about
That,
I've been talking about this.
Yeah, no, sorry. I was looking at a B because again, that was telling pretty much the same story as C and D was,
Yep.
but
Yep.
except
B, C, and D are all related, just like in figure one, right? Is
mm,
uh is C and D as well, right? C and D are the linked. Or and here it's B and D are the same ones.
yes,
What
okay,
is
right,
C in this case? C is 15 recovered in parallel with peptide pools.
right, okay, yeah.
In
So I mean, this is
parallel.
so I didn't quite understand what the lines meant in this because they're matched
They're
candidates.
matched, yeah.
Oh, right, yeah, I think I see now. So they're in one end they're looking at the SARS-CoV response, and the other one SARS-CoV-2, and then seeing like whether they match up.
Mm-hmm.
Uh,
Mm-hmm.
and they actually seeing that the actual responses seem to I mean, for one of them, it seems to be like exactly the same, but
Yeah,
well,
a bunch of them, right? So it's like
yeah, a bunch of them.
if you see straight lines, it means it's about the same, right? Strength of
Yeah.
response between one or the other. But if you see lines that are curved, one above, one down, right? It means that there's less. It's a lower response. So
Yes.
there's a fair number. I would say I can't. They don't quantify it for us, right? But
But
there are almost as many straight lines as I see diagonal lines, at least
yeah.
in NP2. In the NP1, I would say there's more straight lines than there are diagonal lines. And I think I
Yeah.
even see one upward upward line.
Yeah. Well, so like NP2, I think I'd be more confident in saying that the SARS-CoV the responses aren't exactly the same because most of the lines are like swinging downwards, but whereas
Right.
NP1 it's a bit more like there's a little bit more noise there in the story, so I'd be
So
less confident saying
that's
that.
just one
But
of the it's a region, right? NP1
mm,
versus NP2, those are
yeah,
regions of the nucleic capsid. Um,
yeah. So again, this is drilling into a bit deeper and looking at like what exactly going to the basic science end of things.
I wish that
Uh,
they core. I mean, maybe someone more adept at reading this could figure it out. But does this have anything to do with what epitopes match up in Figure two, right? In these pools, do they correlate into the NP1 versus NP2? Like,
uh, I don't. Yeah, I think that they do have some figures that look somewhat into that, but again, I didn't like see anything spelling that out specifically. But that could just be because I'm not great at reading papers. So
yeah, I mean, it's like kind of yeah, it's kind of dense in the sense I don't really know, but I mean that could be one of the reasons, right? Like it is possible that the information in Figure two could help us answer that question. I can't figure it out right now on the spot.
yeah, so I mean, we're all doing this live right now, reading the paper for you in the audience. So uh, you can correct us in the comments.
Yeah.
Um, so I guess this
But yeah, we should talk about E then, right?
yes E
So then they also they do a stimulation
yes. Uh,
to expand the group and also to see whether or not in the expanded group they have the cross-reactive responses. So they still do, right?
Yeah.
Expansion doesn't change what they saw for the most part in terms of cross-reactivity.
Yeah. That that makes sense. Not really much more I can say about this particular
Yeah.
section of the paper. What else is there? Okay, right. So I mean, they also talk a little bit about. So they've got like an extended figure, but that also kind of highlights what they were saying, I think. So extended data figure 6. Again, this is one that I didn't look into too greatly.
Extended figure 6. Let's see.
Yeah. So because again, this was one was kind of. So I mean, this this again was. Where did
Yeah, this is between. This is in the. This is expanded. These are the expanded cells, and it's like it's the detail. It's like instead of just showing, because I think it's ELISpot, right? That's collected in E.
Yep.
So this would be the correlating. This would be the fax plots to go with it. It shows the same thing. That in the expanded group you do see cells that are. Oh, this is like but the two things on these graphs, it's not CD4 and CD8, it's the interferon gamma and TNF alpha.
So it does kind of pull things together a bit between the both immune cells.
Yeah, it's seeing like there's double. It's double weird. Staining. Yeah, there's some double staining in the NP1 too. Yeah, in the NP1 pool. Yeah. It's like it's the same in the other subject, the NP1 pool.
Yeah.
What did they say in the text about that one?
They basically say that let's see, ICS staining confirmed that SARS recovered have memory SARS NP specific CD4 and CD8 T cells. So not being too familiar with the markers for memory cells. I mean, I know that there are some double positive like cells that experience things. So I mean, they go into memory cells in a bit more detail later, especially with the next thing where they look at the people who have been unexposed to SARS or SARS-CoV-1 or CoV-2, which is in so figure 3A. So 3A and 3B.
Yeah, they're gonna repeat. They're gonna repeat again what we saw in figure 1 and figure 1 and figure 3, right?
Yeah, figure 1 and figure 2 basically. So they're gonna like repeat that, but with the people who should not have any responses to any of these proteins because they haven't had any experience with SARS-CoV-1 or SARS-CoV-2. So I think they actually did an antibody test to check whether people had like kind of SARS-CoV-2, and I think looked at like their history. I mean, because I think also like SARS-CoV-1 antibodies stay up for a long time as well, so they may have checked for that as well.
Yeah. I mean, they want to know. Yeah, they had some confirmation that these people hadn't been exposed to COVID-1.
Yeah. So I mean, they basically look into them and they find that actually they do seem to have a response to these like proteins, which that's the real interesting part of this paper, because all the rest of all the rest of the stuff you'd hope for, but this is an unexpected and interesting result, which is what like all scientists hope to find. I think this is where the bit where you go.
That at some, I mean, it's like 50%, right? Like, so some, some of them have, and in that group, like C, right? Figure 4C. In that group, a lot of it seems to be against the antigens that weren't what we saw in the people who recovered.
So that is quite interesting because I mean, again, these are the antigens that are expressed very early on in the infection cycle. So I mean so it's interesting to figure out like kind of why they respond to these antigens.
But I mean, that's sort of why I think that there's that emerging hypothesis that could come from this, and we could ask like a follow-up study could be like, is it because like the infection drives response towards MP preferentially, right? For some reason, like either on the viral side or just like some sort of infection dynamics thing that's going on. But it could also be that in seasonal versus these ones that there's some mechanism inside of
Yeah.
MERS and SARS, right, that drives it towards that level.
Yeah, because they've already established that seasonal viruses have quite a lot of homology in these specific proteins.
Mm-hmm.
So so the next step would be like perhaps that's where the crossover is coming from. It's not because of they.
Oh yeah, I think the other thing I want to say, we kind of didn't talk about this when we talked about the expanded group, but
Mhm.
the expanded data is also sort of telling you how long-lived that response might be.
Yeah. So I mean, there was like a graph in the expanded data that shows like multiple like across. They say retest people on multiple days, I think. So they
Hmm.
retest one person, subject H2, on like the 13th of February 2019 all the way up to 2020 to see whether they've got
Mm.
like reactive uh,
T
cells,
cells,
T cells,
yeah,
yeah.
all the way out.
And again, the T cell response doesn't decrease, and in fact, it seems to increase slightly. But then but yeah, going back to the yeah, so yeah, they in like C and in like they do like have side by side comparisons in I mean, oh gosh, I I've messed up because my overlay's cut off the reference, like the actual like
Oh, where
letter
does
that.
that graph come from? Let's...
Yeah, I'm forgetting. I completely like. So like, because this paper, one thing it has a lot of is graphs and things. I've separated things into subgraphs as well, so
Yeah. Oh,
they
that
appear.
one's extended figure 7. I think is
Yes.
what we're looking
Okay.
at. Yeah.
Yeah. So extend
Yeah, so
figures.
in this, they made this easy for us. This is extracted, right? Basically, a nice way of looking between all of the three types of samples, right, that they picked
Yeah.
out. What the antigen that the recovered serum or the unexposed serum was responsive to, the T cells in
Yes.
those serums. And this is where you can see very plainly, right, that it's bizarre that the unexposed group has so much orange.
Yeah. Yeah. And yeah, again, that color really helps us a lot to like see the difference.
Yeah, and the the follow up again. This is why it's so basic in my ways. Like, there's so many follow up questions. Like, why is that?
Yeah.
Um, but for the rest of the paper, they just continue to characterize, right? Like,
Yeah. So
what
they
is the
showed that they
difference?
expand in D. In
Yeah,
and
so in
they.
D they show the same thing that when they expand it, they still get the same response.
Yep.
They lose it in some though,
Yeah.
like
Yeah.
NSP 13-2. There's like
Oh
there's an
yeah.
equal amount that lose it that had gained, or it's kind of weird. I don't
Yeah,
understand what's going
that
on there.
is
It looks
interesting. Because yeah,
like
that you wouldn't
like
expect that.
under expansion. No, under expansion they get ones that are more reactive, is what's happening, right?
Yeah,
They're going from 0 to something.
yeah,
Which is the point that I was bringing in. Like, part of expansion 2 could also be this idea of, so right, they do in expansion they do that cell sorting, right? The cell sorting technique that we say we should find the cells.
yeah,
There might be no positive cells or very few positive cells in that, but when you expand it, you get a lot more of them,
yeah,
right? And then you run the test again and see if they respond to the pool, right? And oh, they lo and behold, there are cells in that group that respond. So in some
yeah,
ways, you could say that there are cells, long-lived cells in these samples. You just can't see them, right?
yeah,
And yeah, and they end up being ones that respond. So like, because D, it's not just SARS-CoV-11 unexposed donors. Yeah, so they went back into the pool. This is like an
yeah,
11 that weren't previously looked at, right? It's not like these were ones that already got screened with interferon or something else, right? These are, we didn't look at these 11.
yeah,
So it's, again, they're trying to pile a bit more evidence on that, like, for these seasonal coronaviruses, there's memory inside of these T cells, right?
yeah,
When you expand the pools, there's stuff that shows up that wasn't there before. So it could have been like low levels of these cells were around.
yeah. And I guess we can move to D, where they do actually try to look into because they've got like line up of the proteins and have them side by side with the responses.
Mm-hmm.
So again, showing like areas of like high homology. So they've got like they're looking at let's see what protein are they looking at? NP101 to 2020. So this is the nuclear protein, and looking at that homology between different different coronaviruses and looking at the reaction of I think stimulated populations. I think is that yeah
Mm-hmm.
yeah the expansions, and then um, so they look at like the three different proteins like from like so NSP7 they get two like kind of segments as well, and uh, yeah, I mean it's interesting because like looking at the homology lining up, you think the nuclear protein has so much orange there. You'd expect that like there would be a big reaction. And it
They
does.
in E they say that they do a peptide pool matrix. So that's actually going back to figure two. I mean, they don't present all
Mm,
that data, but they did it again.
yeah.
Right? They did it again and they show us the positive with the sequence underneath it,
Mm,
right? Of the peptide that they chose, which is the one that they put in alignment, right? They
yeah.
put the alignment peptide in. But they tracked it down. They said that in these three subjects, this is the peptide that gives the most stimulation.
Yeah.
hmm
Uh,
And those peptides have homology. Well, I mean, of course they must, right?
they
Like,
must do. Yeah.
if they didn't, then the result I wouldn't. It'd be a very different story. Yeah.
Yeah. I mean, it's interesting, like the areas that do have like homology, and because I mean, yeah, it's yeah, I think that I've run through most of the figures by now. So I mean, there's a few more things on. Extended figure 9, I believe, has most of the rest of the stuff, which is a lot more like dusting off some. Uh, so I think like they're. So I'm trying to rush through my things. So they do look at yeah, no, sorry, the extended figure 8, where they try to look into memory, I think. So again, looking into long-term effects by looking at in vitro expansion, I believe. I might be misunderstanding this because uh,
So in eight,
yeah. So we've seen the longitudinal analysis where they collect PBMCs, so blood samples from one person as they go on, and then
hmm
they also like I think they stimulate uh,
yeah.
so
So
they
it's they're this is the long-lived elements, right? That
yeah
they're in this person for a while,
yeah
and now they expand it with that specific epitope that they find.
yeah
And yeah, like what's B showing us?
so B is showing us so I
Activating
guess
peptides are in red and neighboring in black.
right okay
That's just where the peptides are. Oh, they're just showing that it's like it's very specific for this region.
right okay oh
They're
yeah I
showing
see it
us that it's
now
very specific for the region. Again, like this is because there's when you say that inside of unexposed individuals there are people who are responsive,
yeah
right? Like that's very bizarre. Why, right?
mm,
And of course the reason is that they're proposing is it's because of the sequence homology, right?
mm,
And they just are going to show us now with more resolution, right? That it's not this region to one side or this other region to the other side that gives the response. It's only this peptide region here. And again, this peptide region has this lineup. So actually, in some ways, it would be nice to see the alignment in C. I guess in C, that's what they do, right? They give us the alignment of 36 to 50. But is
yeah
that what they're studying in B?
I uh I mean things it looks like they're looking at multiple alignments in B, whereas in C they're drilling down into like
Just the one.
just
Yeah.
one. So yeah, I mean again I think they're showing that this one is actually very specific to SARS-CoV-2 by the looks of it. Um, but
56 to
so,
60. Oh yeah, no, they're here. 56 to 60 is here. Yeah, 36 in H3. Oh, that probably is the one. In figure B, they have subject H3, and on
yes,
that graph they have the 36 to 50 segment. That's the one that that's the one that they're looking
yeah, yeah,
at the alignment there. H21
yeah.
is 36 to 50 as well. So H21 is the same fragment.
Mm, okay.
H7 is a different fragment, and H2 is a different fragment as well.
Yeah.
Yeah, so
Hmm.
H21 and H3, they just expanded down there to show us the actual alignment. Um,
Yeah. Um,
and
so,
that it's done in with ELISpot ex vivo.
hmm. I mean, I, I mean, isn't all that spot done ex vivo? It'd be quite difficult to get someone to
Yes,
swallow one of
yes,
those kits. But
yes.
yeah.
You know, all the ELISpot was done with ex vivo. It's also because
Oh,
it's like that's closer.
I guess like ex vivo, like grown cells or something. Rather than a direct sample, maybe cells grown ex
No,
vivo.
it's weird. I mean, what they're showing in C is that they stimulated just with the SARS. They stimulated with the version from SARS-CoV-2, but not the other ones. And it's
Right.
SARS-CoV-2 that it's specific for, not not the other ones.
Right.
Yeah,
And
this is going. They're doubling down on the surprising aspect
yeah.
of their finding, trying to really surprise us that it's not actually the homology regions that
Yeah.
drive the response.
Yeah.
I don't know.
That's
I'm weirded out about this. Like,
yeah.
I guess this is why it's in the extended, right? Because it's
Yeah.
like they would like to show us this thing that is really strange, but it's also that I don't know, like maybe they have to do some more different lines of evidence or bring us some theory as to why they think this is happening. Why should unexposed people have specificity for SARS-CoV-2 and not any of these other viruses? Because originally I was thinking that it's cross reactivity. Like originally when we see figure where they first do original SARS.
Mm.
Yeah, sorry, I don't think I paid this much attention to this extended figure
Yeah.
before, and now that I have, I find it very bizarre because if we look at figure 4A, right, like that's where
Mm.
we first hear the surprising news that there are cells inside of unexposed serum, right, that have specificity for SARS-CoV-2 things.
Yes.
And the first thing I would think of is let's then remember extended figure 1 where there's homology. Those sequences must be because of the homology between the two, right?
Yeah.
Must be.
Yeah, it must be.
Uh, but in extended figure 8, they're sort of trying to tell us it's not because of that.
Yeah, it's
Right? Like when
yeah.
you track it down to this one peptide that has the response, and then you give the peptide in the sequences of SARS-CoV-1 and the seasonals, it doesn't give the response.
Yeah.
Yeah,
So
they're trying to corner my surprise
yeah.
to tell me, oh, by the way, it's actually very surprising.
Well, I mean,
Okay,
sometimes data
weird.
doesn't follow a story, and you try to make a story, but you don't want to like let the people. So you like have things in the extended data that don't necessarily fit your story, but might be important for future generations, so that they can pass things through. And
Yeah, I mean, this would sort of be something that I would want to see. It's like the beginning of another story potentially,
yeah,
but it's not taken far enough to tell me why. Like a follow-up would be to see this again.
yeah,
um, in another figure. Like they showed us in H3 and H2, which is specific for just the 36 to 50 peptide in NSP7. But what about what's happening in H2? Right,
yeah,
that's the NP amino acid residue. Is it the same thing there? Is
yeah,
it right? Is it? Yeah, I think that like again they drilled down super specific here,
yeah,
but at a cost of so with the benefit of showing us something very very surprising in a specific instance, it's also only in two people with one peptide from one of the things, right?
yeah,
Yeah. So maybe showing us in another way, that would be the way to restart the story and tell
mm,
us another story, right?
yeah,
But yeah, I guess that's why it's in the extended figure because it really fights against your thought about it.
yeah. There's also so figure 9, which is where they start to look into more in the memory aspect of things, because I think one of the reviewers made this point that it could be that in our blood there are like cells that haven't encountered the receptor that is going to be the one for them for a
Hmm.
while. So the theory was that maybe this is just cells that have never seen this antigen before reacting and learning it from there. and then
Yeah.
based on that, and therefore it could be
Yeah,
you
like
might not.
maybe for whatever reason this antigen is like it hits a subset of like hyperreactive expanders, right?
Yeah,
Because like the again like the hypothesis of seeing the expansion is supposed to be like, oh, it's like memory, it's like, oh, they remembered. But I guess there's a counterexample that did they even, or the counter question would be that are they even expressing the molecules that we would associate with remembering?
yeah. So we have to make sure that it's not just like the first time and then suddenly, oh, you're getting a response, but that's because you get a response anyway. It doesn't necessarily indicate memory. So trying to make sure that we have that evidence there that this is because people have been the immune response is due to something that previously happened. Um,
Yeah. Well, they only show, and so they do that, right? They try to do the panel where, I mean, that's C,
uh,
where they're
yeah. So in C, they basically they do that panel where they look at some see some
markers of terminally differentiated effector. I mean, that's what it says in the figure
yeah.
text, or in the text of the paper, right? They're markers of terminally differentiated memory cells, so not naive.
Yeah. So I mean that's
And
I
I
mean
think
again
also the fact that I think that they're supposed to also suggest that in B because they're also very specific for a cell for a certain type of presentation.
yeah. Yeah, so I feel like because this is where we're a bit about. This is very immunology and thing about. I don't remember the exact
I looked up
clusters.
I looked up like what it meant that it was HLA restricted. Uh, from my understanding, like those subtypes A, B, C, and those numbers, like they're different between different people.
Hmm,
That was my understanding, right? We have
right.
different MHC class subtypes.
Yeah.
And so what they did essentially in B was they showed that this person has this subtype, has
Right.
this haplotype, right? Like
Hmm.
this is the haplotype of the person that was infected. So
Okay.
I actually don't know if that right. Like it doesn't really make any sense to me. Oh, I guess it's it means that they're not reacting just to I guess what is known here what's good about B is that that is the antigen presented in its correct context.
Hmm.
It's
Yeah.
loaded onto an MHC. So it's not just like spirulous peptide,
Hmm.
right? Like that floated around and did the induction.
Yeah.
It's like loaded into the MHC molecule specifically. So yeah, I think that like that's what we're getting from B. Not I feel like the restricted idea that's not what they're showing here. I think that idea that it's like a biologically relevant context is maybe the important part.
Yeah. So I think we've like maneuvered through most of the figures that they're presented in the paper. Um,
Yeah, so like what do you think you learned after this? You know, like what are you going to take away and remember from
I
this
mean,
because
so the interesting thing about this is there's a story emerging about herd immunity in the news. I don't know if you've heard about this about how herd immunity might be higher in the population than initially expected, which is why because I think a lot of countries are recovering from COVID a lot earlier than they thought they would. That
Mhm.
like the and maybe that's because of the delay in actually between like exposure and symptoms coming up. But there's also this alternate idea that
Recovering how? What do you mean
as
recovering?
in for like the number of infections compared to like how lockdown is being lifted, the number of infection growth doesn't seem to be as high as people were
Gotcha.
expecting. But that's
Yes.
partly
Yes.
because again the gap between exposure and symptoms showing and gap between symptoms and death. So
And also that like it's in the news, behavior has changed. It's not
yeah,
that like we're behaving in the same way across the board, right? Like I think that like that's a really important covariate
yeah.
there as well. Okay, anyways,
Yeah,
I just
people
wanted to
over
throw that in
yeah,
there.
I mean people often overemphasize the molecular aspects of a disease when actually behavioral stuff has a really big important factor.
Yeah. Yeah.
Um,
But no, please link it back. Tell
so
me I interrupted your
linking it back.
story.
So like, there's also like this other story about how the antibody responses for COVID-19 aren't necessarily reliable for testing.
Hmm.
So one things that the question I was hanging in my mind is like, how do we really know that these people are entirely naive to COVID-19?
Hmm.
And are we actually seeing people who have been exposed to it but managed to manage their infection in such a fast way that didn't show symptoms? But
Sure,
that's
sure.
but again, this is a theory coming out of cloud cuckoo land from my head. So obviously like
No, but like the data that is displayed in this paper could be something that helps support that story, right? Like you
Yeah.
could say that, oh, maybe the reason is because we have some protective cross immunity from previous exposure, right,
Yeah.
to this or that.
I mean, the researchers do like put forward this idea of like an exposure from like animal like hosts for like a whether with defective virus particles. So they get to some point, but they only get to the early stage of infection. So that's why they react to only the early stage expressed proteins.
Hmm. Hmm.
And I was thinking of the idea of perhaps for COVID, like because again, if you have T cells that react to those early expressed proteins, then they're going to be very very sensitive to early on in the infection
Yeah.
cycle.
Hmm.
So
They would
it,
expand quickly, and it could, again, as I said, like even cytotoxic T cells could have feedbacks into the humoral immunity response,
yes,
right? It's like I mean, I think often in immunology people talk about like branched responses and how it goes down, but then like the more people study things, there's like weird crosstalk mechanisms, and like
yeah.
so that could be it too, right? Like yeah.
But I
um
mean, I guess the main thing is like the homology of the immune response, the kind of diversity uh, in how like coronaviruses behave towards the immune system, and I guess like in getting an idea of like the T cell response to the coronavirus and what the things we need to consider there that might be different from the antibody response, because
mhm.
the idea of
Yeah,
like
well, I mean,
yeah
certainly from the consciousness perspective, I do agree that like one of the reasons I liked what I learned from this paper, which isn't really about the paper itself, right? But is just that there's so much talk about humoral immunity, and again, for good reason,
mm,
because in studies in SARS and MERS, it was shown that neutralizing antibodies are like very important in differentiating severe versus mild disease, right?
mm,
But there's more than that in our immune response, right? And like outside of learning from this paper, but just like reading the paper in general, it's refocused my attention, right, on the idea that like, oh, the immune system's a big mess of things, and like here's another element, right, that we haven't been talking about, right, the T cell immunity. But much harder to measure because we're
yeah
not. This is not going to be a this is not going to be a lab test very quickly. Like these research labs need very specialized equipment in order to do these things, and like they take a lot of care with their samples and expertise in handling cell culture.
mm,
So this is not something that will just like appear for many years, right? Like if diagnostics appear at this level, it won't be until we figure out some other technologies first.
yeah.
Um, yeah. What am
So,
I? I'm trying to think of what I think it's bizarre that yeah, like I'm struck by that difference between the immunodominance of the early proteins in NSP in uninfected people and then the immunodominance of NP in recovered people.
yeah.
It makes me think you're right. Like you said, the authors hypothesize about like a like early aborted infection or something
Mm,
like that versus late aborted infection. My hypothesis would go down the lines more of um, uh, like actual pathogenesis, right?
mm,
Like in the course of pathogenesis with SARS and SARS, MERS, and COVID-2, like that's a more severe disease. Like maybe that pathogenesis is pushing the response to NP2, right? Those antigen pools instead of the NSP. But again, I guess that's the more complicated hypothesis than the one that just early abortion,
yeah.
right? Yeah, that's like kind of too complicated maybe for I like the way that they said maybe it's just early aborted cases. Um, I mean, if you could just it oh yeah, but like early aborted, but they could be early because they made that hypothesis on the back of I'm not sure that it's only specific for SARS-CoV-2. Hmm.
So it's
Yeah, I'm not sure. I guess I'm a bit. I don't know what's the better way of thinking about that. Then this figure eight has really thrown me for a loop. Like
yeah.
like why is it important to show that in these unexposed people it's this antigen from SARS-CoV-2?
So
I really don't know.
yeah.
Anything that I thought about this paper has kind of been convoluted through this piece of data. Like I'm choosing not to think about it mostly.
So, okay, so I'll see what they wrote about it because I think they said that like the low homology peptides covering the sequences of common cold coronaviruses failed to stimulate PBMC in the NSP like So basically, I think the thing they're saying is that the common cold viruses aren't necessarily the way to explain this prior immunity.
Yeah,
So
exactly. Yeah, no, that's what I mean. I don't,
yeah,
but like, what is that? I don't know how that fits into the story of, right, of T cell immunity, it being important, or if it's important to these diseases,
I mean,
right?
yeah. I kind of like agree with you there because I feel like they're also kind of stretching when they say that there's like an unknown like animal like coronavirus that doesn't appear.
Yeah, yeah, they're really trying very hard to figure out to explain figure eight, and I guess to me, I just like I want to say it's only two people and one peptide, right? Like I don't think that it speaks to
I
um
mean, yeah.
it,
I'm
it, it doesn't speak to like a greater knowledge of the population and how it interacts with SARS-CoV-2.
I mean, yeah. Personally, my opinion is I think that it's more plausible to explain that they were infected with SARS-CoV-2, but they didn't show any symptoms, and they
Yeah,
didn't show
yeah,
up on
like
tests.
right, there we go. Like what about that? That's a great one.
That's
Yeah.
but the problem is that's nearly impossible to confirm because how do you test? Oh, we test T cells. Oh wait, and then you get in
Right,
a
right,
cycle, and
right,
you can't. So it's like the disastrous unprovable hypothesis where we need to have like an entirely need to go back in time and then see whether they
right,
get live virus. So
right.
it's all
Yeah,
look
like
for.
that. It's too like figure eight has become too extended figure eight became too surprising for me, right? I'm more interested in thinking about extended figure seven, which is again related to in the article. It's the same thing with orange.
Yes,
Is figure 4C, right? Like extended figure 7 is just seeing figure 4C arrayed against the other ones where we see the
yeah,
dominance, right?
that's nice
Like
and clean.
yeah, I mean like at least there like I'm like okay, so we know that there's some reactivity in these people, right? And like correlating that with D that like it's not spiraless, right? Like when
Yeah.
you expand those populations, it's there. Like some people react for some reason to this. I'd rather start thinking about why here. Like to me, this is already confusing enough. And to dive down there and then have to explain that, I don't know. It seems tough.
I mean, I like
Uh,
that there is. I mean, I feel like every paper is going to have something that is unknowable, unexplainable, because
yeah,
is,
for sure.
and I feel like that's important to acknowledge that nobody know, and that's actually what makes this paper really interesting for me. But that's why we can actually spend quite a lot discussing it because we've got this mysterious result with multiple explanations.
No, I mean, I was so enamored by this paper. That's also the reason why I kind of want to discuss it more because, like, after getting through the methods, if people can understand flow cytometry, right,
Yeah,
and the ELISpot pools, and then the difference between ex vivo and expanded, like, there's such a strange set of results here, which again were done in humans,
yeah,
right? Like these are like human responses, small groups of humans, but like humans nonetheless. And
yeah,
I find that fascinating. And I think it's important to also realize that like figure 4B, right? That's only half of the people, the unexposed people that they tested,
yeah,
right? And so like yeah, and in that half it's like really actually quite heterogeneous. Like yeah, a lot of it's NSP and NP, but like yeah. um
yeah.
People also responded to the NP. It's like kind of like a crapshoot
I
of
mean,
people.
the thing I'd be very, very interested in, from like a vaccinology perspective, is whether there would be a difference in outcomes if we looked at like vaccines based on the NSP versus the nucleocaps, the other
Right.
proteins.
I know that. I think like, that's why I think I like this. Yeah, I'm gonna stay on figure 4 as the sort of jumping off point to like
Yeah,
think about other things and how it impacts stuff. Because you could also say, okay, regardless of how it happened, right?
mm,
There is some sort of response, right, to
yeah,
some of these antigens. So what does that mean for for vaccines that get developed against these targets? The live attenuated vaccine, which has the
yeah,
whole slew of proteins in it, right?
yeah,
What is it like? There's a heterogeneous population of people out there, right? Already, that's what I'm definitely confident of saying, right?
yeah,
In the community, there's heterogeneity into responses towards these non-structural proteins and nucleic capsid proteins in our population already, right?
yeah,
So if we go for strategies that have these proteins involved, right, or are somehow dependent on these responses, right, we're gonna see variable responses.
yeah.
Um, Yeah.
So, I mean, I'm looking at a lens of like what I mean from my lens of my crazy theory that they're all these people have been infected, but what some of them controlled it really well and didn't show any symptoms, and some of them did. In which case, it might be
Oh
just
yeah, yeah. I'm not
so.
seeing it through that theory. I'm saying
But
like
yeah,
it is some sort like I don't know through what mechanism, whether it's like exposed before from another
yeah,
virus, right? I'm just saying from the like
yeah.
what I'm
Actually, you're
learning
right.
from it is that like it's a consideration when you go and you vaccinate people, right? That people have had some cross reactivity before to
Yeah,
something else, right? And that's going to change the immune response, right?
yeah.
So
You
so yeah, so like that's
So
interesting from that
that yeah,
perspective.
that's actually because again, for when you're looking at control in those populations, if there's already a background like cross reactivity, then suddenly a vaccine that is effective might look less effective because
Yeah, yeah, or it just might make a different like some people won't respond and maybe this is a reason why the response is different, right? Because they went down a different path. Right? Because like, this is also not necessarily the humoral immunity path. This is the cell-mediated immunity path. So maybe that's something else that yeah, that's going out there.
yeah,
Maybe in some ways that means that spike is good because it just bypasses that. Like we're
yeah,
just making spike neutralizing antibodies. Um, but again, like if that's not the protective factor, because we still haven't seen where this
yeah,
is still like we're putting a lot of hope in the fact that spike neutralizing antibodies is the way that we're going to get
hmm,
protective immunity in people,
yeah,
right? And again, because best evidence shows that that would be the case. But if it doesn't turn out to be the case, this would be amazing information to have when going to the drawing board,
yeah.
right? And like, I would like to again, like, it doesn't say a lot about what's protective in this paper, right?
Oh,
So yeah, so
yeah. It
talking
can't
about how
really.
that correlates, yeah, it's not talking about protection, but it does talk about what people respond to,
Yeah,
right? Like what's in people's bodies,
yeah.
maybe,
I
right? What
think
could be in people's bodies?
yeah, that's something that I think is very we need to be quite clear on. That they aren't they're talking about two very specific proteins and they can't necessarily
Mhm.
extract this. They even though I have made those sorts of exclamations, I don't think we can actually really extract this to actual immunity because again
Well, they never
they
look at any sort of protection at all,
yeah,
right? They're just looking at reactivity of the cells.
exactly.
Have the cells seen it before? Are they responding in a way as if they've seen it before?
Yeah, I mean, if they were looking at that, they would have to look at all the proteins, and because they miss out spike completely, and spike is something that is very important on everyone's minds when it comes to
Yeah.
this. But they're looking
I
more in.
mean, and they would have to have some other assay. Like I don't even know. Like how do people? I tried to figure that out when I looked at the two papers that had shown this before to have correlate with mild and severe disease. But like that's how they showed it. They
Yeah,
showed just the correlation of these cell types with mild or severe disease. So like there's no more mechanistic assay to go in and say like these are the cells that are going to do the protection, right?
yeah,
It's just like a correlate with mild or severe. And so like yeah, I don't think that that information is open to us right now,
yeah,
like with the sort of the assays that we have.
yeah. So yeah, I mean, I think I've said all I can say about this paper without. I mean, I've already delved slightly into Fantasyland with my crackpot
Yeah,
theory,
no,
so I
I think
mean,
it's good. I think
yeah,
that it's good discussion about it. We took a bit longer today, but
it's a big
I'm glad that
paper.
we did. Yeah, it's a big paper. It's a
Yeah,
big paper.
I mean, yeah, it's different from like the vaccine papers, which have like three figures, and they're basically saying stuff that we already kind of know. We have to. This is telling a basic science story, so putting together a lot more problem solving here, rather than
Yeah,
like yeah.
yeah.
So that's kind of why it's interesting to look at these kinds of papers alongside with does Moderna work? Does
hmm
Chaddock's work? This is
hmm
kind of looking into like the underlying factors that and like as
Yeah,
microbiologists we're kind of interested in how these things work. We're not just interested in like what they can do for us, but like
yeah.
yeah,
I want to, I'm itching to see a paper done in the mouse model that talks about pathogenesis, right? Like,
yeah,
because right now the mouse model is poised as a way to look at a vaccine or like,
yeah,
like poised the way to look at like preclinical efficacy data. But like, where's this paper with the mouse? I mean, I don't want to see immune actually. I don't think that's, yeah, actually, no, I do. But I want to see it like on the virus side more, right? I want to
yeah,
see like the viral antigens being varied in a certain way, producing a different disease state, right, in the
yeah.
mouse. right? And then taking that information and then making a hypothesis onto the human population. You know,
Yeah.
that's what this paper has whetted my appetite for, right? Some basic pathogenesis research with this new virus.
I mean, we could actually try and look at it. So next week we're going to be looking at a whole bunch of scientific papers that have been released, and we can delve more about that question, look into more pathogenesis.
Yeah. I'll definitely have my eye out more, I think, for that after reading this.
Yeah, so if you viewers want to like check that out next week, then we're going to be out discussing a whole raft of papers, giving our like first look like ideas on papers before we pick one to drill down in detail like we're doing here.
Yeah, yeah, absolutely. Yeah, well, we're really happy you guys could join us.
Yeah.
If you've made it to the end of this probably monstrously long video now.
Yeah, I mean,
Yeah, please communicate over Twitter. We're looking for suggestions to help form the way we speak about these things.
and if you have any like things to add to our discussion, we'll be picking up next week if you've got. So you can tweet me if I've got many things wrong and correct. We want people to nitpick us because who wants a head full of nits? I mean, yeah. So yeah, it's been wonderful having you here. Thank you so much for joining me, Danny. It's been great hearing about all of this. So I think it's goodbye for now, and I'll see you next week.